Hi, welcome to one of my tutorial videos. Now if you're watching this particular one, you're curious to know about polymerase chain reactions, also known as PCR cycling. Now, if that's what you're here to learn, let's get started. Cycling. Now, PCR cycling is a process where a specific region of DNA is duplicated into millions of copies through a series of heating and cooling processes. Before we begin the actual PCR procedure itself, we need to prepare the 5 primary ingredients that are necessary for PCR to work. First, you want to start off with your particular DNA sample strand. Now this strand is just the region of DNA you want to be copied. Then, you have your primers, which are just small fragments of DNA. Next, you have Taq polymerase enzymes. You specifically want the Taq polymerase enzymes because these are thermostable and able to withstand the fluctuating temperatures that are going to occur during this process. Next, you have DNA nucleotides. These are just your 4 simple bases: adenine, guanine, cytosine, and thionine. Lastly, you want a buffer extract. Now this buffer extract is key because it helps to ensure and stabilize the right conditions for the reaction to work properly. Once you have all 5 primary ingredients together, you want to collect them all into a test tube where your copies of DNA will be produced. Now, PCR uses a thermal cycling process that consists of a method of heating and cooling sequences, as it was described at the beginning of this video. The procedure includes 3 core stages: denaturing, annealing, and extending. To begin, we'll start with the first stage, which is denaturing. During denaturing, start the PCR cycle with our specific DNA sample, as seen here, and heat it up to a close boiling point of 95 degrees Celsius. The high temperature will cause a double-stranded DNA to separate into individual strains, hence the name denature. Then, at the anneal stage, the temperature will drop to 60 degrees Celsius or even lower. In this state, the DNA primers, as seen here, are able to secure themselves onto either side of the DNA strand template that are complementary to match its sequence. Here. Once the primers are fastened, we begin the extending stage when the temperature is slightly raised to 72 degrees Celsius. Here, the Taq polymerase enzymes go to work and bind to the primer sequences that are attached to DNA strands. The enzymes then add nucleotide bases to the original sample fragment and extend the copy of DNA so there are new links of replicated DNA. So, we're going to start with this first DNA strand. As the Taq polymerase enzyme goes up, the nucleotides are added so it complements the original strand of DNA, as you can see. Then the same thing goes for this DNA strand too. As the Taq polymerase enzyme moves down, more nucleotides are added. After the extending stage, the first cycle of PCR is complete. Then, the 3 stages of denaturing, annealing, and extending are repeated at least 20 to 40 times, where the number of DNA copies are doubled each time to result in millions of duplicates. So after 2 cycles, there would be 4 copies, and so on and so forth. Now this process could take 3 to 4 hours, depending on how many copies you would want to be produced. When all the PCR cycles are complete, each strand of DNA is verified to make sure the PCR worked by the method of electrophoresis, so we can check the quantity and size of the produced DNA segments. Now that we finished our run through of the PCR process, you know a little bit more about polymerase chain reactions. Now, PCR is very important in the medical field because it could help identify dangerous pathogens that are the cause of diseases. Not only that, we can study a patient's risk for certain disorders by examining mutations in their DNA sequence that match the others of the disorder. Now that we've finished, I hope you've enjoyed this tutorial.